Hello, I'm Dilara Diken, and I'll be talking about my project that I conducted during my OmicsLogic Research Fellowship program. And the title of my project was Radiation Threats to Humans in Space and an Alternative Approach with Probiotics. My project was focused on space-time radiation, a type of ionizing radiation called high LET radiation. So scientists at the ISS was copious amounts of this radiation during over 6 months. So one way to this radiation study is through the microbiome. Since our gut microbiota has effects on our cell and other organs, we can gauge the effects and treatments using this method. To study this, bacteria have to be identified and the best time for OTU So my data set that I collected from it was contained 80 samples, and which were split into two categories, based on days and radiation. So 10 days and 50 days. And I had 4 types of radiation ranging from 0.1 to 1 GY. So there was 0 GY, 0.1 GY, 0.25 GY, and 1 GY radiation. The outputs we generated using the Data to final pipelines, we got the Shannon index graph, and the Shannon index helps us gauge the ENF of the number of species. As you can see on this graph, in the results section, figure 2A, we can see that 0.1 the number of species has decreased. The figure 2B graph, which is the phylogenetic analysis, and you can see, especially for OTUC 4 and 5, the abundance of OTUs has decreased, especially for 0.1 and 0.25 GY at 10 days. So to confirm these results, test and in the ANOVA test, it's reiterating the results from the phylogenetic analysis. I found out that OTU3, 4, 5, and 8 have decreased abundances for 0.1 GY at 10 days specifically. And I also found out that there is no correlation between the days and the amount of radiation that these mice were exposed to. There is low than in control group. Then to find which species these OTUs belong to, I used the JGI website to identify my OTUs, and I found out that they belong to Lactobacillus and Bifidobacterium species. These two species have protein coding genes in the GABAergic pathway. GABA, which is gamma-aminobutyric acid, is the chief neurotransmitter in the central nervous system and has effects on depression and anxiety. Since the enterodendritic nerve and the vagus nerve connect the gut to the brain, we can presume that a decrease in the abundance of these two species, Lactobacillus and Bifidobacterium, will cause a decrease in GABA, and in return might be a factor in depression. And as we all know, one of the common disorders that are found in scientists when they return from the ISS is depression and anxiety. Conducted with long-term exposure to radiation and the synergetic effects of methylreacting radiation together. To conclude, the solution that can be used to overcome this, however, is quite simple and non-invasive, which is using probiotics rich in Lactobacillus and Bifidobacterium. So I would like to thank Elia Brodsky, Dr. Mohit Mazumder, and Dr. Harpreet Kaur for all of their help and being my for being so kind during my research fellowship. So thank you. I hope you like my project and have a nice day.